So, the use of RNA vaccines to control the human brain, explained by Dr. Charles Morgan, a neurobiology expert from Yale, DARPA, CIA, who gave a presentation in 2018 to students at West Point Military Academy. He talked about the ability to manipulate DNA for mind control purposes. Dr. Morgan shows and explains to the elite students of the U.S. military how the technologies known under the acronyms CRISPR, clustered, regularly spaced, short, palindromic repeats, and DREADS, designer receptor exclusively activated by designer drugs, can design anything. So Dr. Morgan shows how DREADS can be infused into human DNA to control our behavior. Designer receptors, which are only activated by designer drugs, can create new cells. They can create new memories and behaviors in subjects. You can also use it to erase or even change your memory. CRISPR technology can be used to only kill certain races of people through DNA editing. So he tells students at Modern War University at West Point that the technology called CRISPR makes a number of things instantly available. You can design anything. You can design a single thing that would only kill one. What you do when you make plasmids and insert them into cells, it sends a signal telling what part of the DNA should unroll, unfold, and produce a product. Product meaning a certain cell, as an example. That is the future of medicine. Dr. Morgan asks in the video, what could you do with this technology if you were in the security and intelligence community? The cells can be designed for specific activities and purposes. They can be placed strategically. They can be controlled remotely. So these cell controls can activate things in other people's brains. We are seeing unprecedented behaviors in our fellow humans. And we really do need to wonder, are they being controlled? So this video, and I will link to it, I'm just going to play a little bit of it and then get into more information about CRISPR technology in particular, which is used in the COVID injections. They're not vaccines. CRISPR injections. So this is Dr. Charles Morgan on neurobiology and war.
The second field that people may or may not be aware of, I always tell my students, I said I wasn't around when they developed atomic weapons, but Dr. Ventnor's work is, my view, the equivalent of the development of nuclear weapons when you realize that he created life in a cell back in 2010. I don't know if people are familiar with his work, but this technology paired with something called CRISPR, which is like an editing software for genes, makes a number of things immediately available. What he did is he programmed yeast cells to produce anything he wanted. They can produce perfume, they can produce petroleum, they can produce any peptide, anything we program the DNA to do, and it's in the living cell. Right? So in medicine, the goal in medicine now is to be able to do designer medicine and therapy. If we can design a cell to get into your body and release the right product for you, you won't be losing half the drugs you take through your liver when you swallow a pill and it gets digested. These can be inserted into you through the hypospray needles, almost like Dr. McCoy on Star Trek, giving you the hyperspray. It just blasts now plasmids into your squamous cells. But Ventnor was able to do that and has the patent on the technology. But you can engineer anything. You can engineer a unique thing that would only kill one person in the world. It's how it's done. You put in a specific gene slicing, You program what you like, you put it in the cell, and it can reproduce and make as much as you like. For those of you who don't know, your DNA is usually all wrapped up in tight little coils. And so what you're doing is when you create plasmids and put them into cells, it sends a signal and tells which portion of the DNA should unwrap, unfold, and produce a product. Now, this is the future of medicine. When you look at this technology in medicine and say, this is going to be done to help people, right? We want to be able to give them medicines. We actually want to correct for genetic deficits. If a kid's born with a genetic anomaly, with the CRISPR technology, the feeling is we can create the portion of the gene they're missing and go have it spliced back in. And that may help a child, either if it's in utero development or once they're older, to have the missing substance actively produced. What would you do with this if you were in security and intelligence? Well, you can do a number of things. You could decide, if you make this gene, we know that certain people in the world who function at very high altitudes very, very well do it because they had a special mutation in their genome that we don't have because we didn't grow up in the Himalayas. But they can function at very high altitudes. Could you give this to people who are going to have to do warfighting in high altitudes and they don't require extra support? Their body makes a much more efficient use and can work under conditions of lower oxygen than the rest of us. You start letting your mind wander. Can it also produce a substance that lets you function longer underwater without oxygen? But these are run by certain mutations in genes, and with CRISPR we have the ability to actually make these and see what happens when we give them to animals, non-human or human animals, that don't have it naturally. You could have the Forrest Gump gene. You guys have been tracking. There's a gene that just makes you stronger. I would say that most of this technology is probably going to be employed by a state and not non-state actors because it's quite technical. But I say that with a caveat. When we studied Shinrikyo, if people remember, they had both uranium mines and regular laboratories where they experimented on both animals and had a whole series of laboratory experiments to develop the different kinds of gases that they wanted. Their goal was to actually mine uranium and probably come up with their own version of a nuclear weapon. But they recruited scientists, PhD level folks, and their goal was to be the rightful people running the country of Japan. But we can't assume that just because they're non-state actors, they will not make use of some technology around this. Related to this is an idea called DREADs. These are designer receptors that can be remotely controlled. So think about it for a moment. You can create a designer receptor, you can create a cell, you can put it somewhere in the body, and you can remotely activate it when the brain's exposed to the right signal. Using this technology, people have been able to transfer memories from one fruit fly to another by signaling through a light stimulus into the retina. Right now, in most animals, it's done by putting a substance into their body that will actually activate the neuron in the way that you want it. So you have the capacity to create any product. As long as you know the DNA sequence, you can insert it into a living system, and you can remotely control it. So in medicine, we think about how we do that to help people, how we do to repair deficits. Other people are going to think about how do they do it to expand possibilities. And one of the challenges that we have is that when you create a cell and you put it in somebody's body, you have to figure out where you want it. What if you want it in their brain? Right? If you want it in their brain and you can't figure out, you don't want to do surgery to plant it in their brain, if I want a product produced in your brain that may affect the way you think, the way you act, one route to that is through stem cells. If you're a quick brush up on your biology, stem cells are cells, they call them God cells. They can turn into anything. They hold the potential, unlike other cells in your body, to become anything you want them to become. And they can go find their home in the body and park there and do the work that you'd like them to do. You can infuse them, and they will find their way into the brain. So once you know that the technology is there to edit, splice, and program a cell, and the technology currently exists to administer it to somebody and have it go park anywhere you program it to go park, proliferate, and do its function, you can have things activated in other people's brains. So if you take these 3 key points, hopefully you can see it opens up a number of both alarming and exciting possibilities. You can have the timed release of information on demand. Hopefully when I mentioned the word CRISPR and word editing and creating molecules with CRISPR out of and playing with DNA, some of you thought encryption and encoding. So DNA encryption, there were I think 8 articles published by China in the course of 3 years, in the last 3 years, and it's quite important. The coding system, DNA steganography, I'll just say the short story on this is people have figured out how to hide imagery in the DNA of bacteria, and when you phosphoresce the bacteria, you can discover the information, or you can have those are just to remind me, you can have the information reproduced in a string form as a form of a protein. Dr. Church up at Harvard has shown quite well that you can store a lot of information in 1 gram of DNA. It's essentially, that many iPads in 1 gram at room temperature. No supercooling required. DNA is highly stable. It's been around on the planet a very long time. So between CRISPR, the storage capacity, and programming cells, the new way to hide information is going to be in DNA. The commercial application is going to be a bit like on Star Trek years ago. Why would you have a digital system when you can have a DNA system? You can store all the information you'd ever need, records, photos, anything. It's simply another way of storing information. It had just been so slow up until 5 years ago, it wouldn't be thought to be practical. But it is. This is the first experiment showing what imagery you can hide in bacteria. This is the latest. It's a GIF file. It was actually programmed into the DNA of bacteria last year. The bacteria reproduced, and the offspring from the reproduction cycle would still produce this movie. Pretty cool. You can hide information in bacteria, and when the bacteria multiply, they can go into a spore form and last for a very long time. No one can scan you and find a bacteria. We don't have anything that can detect that. People, So if you want to be able to encode information, take pictures of information, create something in DNA and don't want it in your own body, it can be bacteria on some portion of your body, right? All they have to do is scrape it, let it grow in the Petri dish, and unpack the information. This is all available now. This isn't science fiction, but you can encode movies. Well, this is what the Chinese are doing with DNA. So in your own neck of the woods, you can begin inquiring. We are doing things with DNA as well. But the Chinese are fairly convinced that DNA encryption and encoding will be one tremendous challenge even for quantum computing. So this is where the race is right now, trying to merge quantum computing with what you'd call a wet hard drive with DNA. Merging DNA systems with quantum computing will be...
Let me just interject here. China is also in the process, or probably has already successfully been able to use CRISPR, these dreads, to create super soldiers. Do you think we're not doing that? US of A? We are. So I just want to play a little bit more how they can erase your memory, inject new memories. They can literally, with this technology, just take a life and inject a whole new life into that human being, and they behave in accordance with that new life, with those new memories, that's why this technology is so dangerous.
So, what's to do with memory? In medicine, we think of memory as a potentially harmful thing when people present with post-traumatic stress disorder. They can't stop thinking about the thing that's creating emotional distress. It's a very active development in the field to figure out, can we erase memory? Can we modify memory? Can we change memory? The short answer is yes. Several years ago, with the PKM Zeta data out of Duke University, this was the first time that anyone had ever demonstrated that if you wash an area of the brain called the hippocampus, it's an area of the brain that's crucial for forming short memories, spatial memories, and then facilitating the transfer from a short-term memory to something that's more permanent and stable over time, that he could train the mice to run the maze, document the number of trials and errors, and then flood their hippocampus or expose it to this, and the memory would be completely gone. Meaning when the rats or the mice had to learn it over again, it was the same number of learning trials. And there was no trace of the memory left. Now the good news for us when we study rats and mice is we put electrodes and cannulae into their brain and can directly affect that area of the brain. If you wanted to poke your own hippocampus, you'd have to stick your finger through your eye and go right back in there. Sounds impossible to get to. Not if you program a cell to go there. So if you decide you wanted to program something that would selectively release PKM zeta, after your meeting with someone, they probably would have no memory of it. That's what's happening in the rats, right? So the technical challenge right now is, how do we get a cell in there to do that in human? I can assure you they're working on that in non-human primates right now. How many, what's the point specificity? Can we get it in there close enough to the hippocampus? Will those cells start reproducing in the next day, make enough of that stuff to wipe out a memory? Related to this, once you start thinking about memory, are chemicals that not only wipe out memory, but chemicals that enhance it. So if you want a better human camera, a better, an individual who can just go see and remember everything, that's the direction that the research in this lane is taking to help people with Alzheimer's. How to give them memory back. So what's being actively studied are the few people on the planet who have hypermnesia. In other words, they remember everything that's ever happened to them. We're actively trying to understand how to unlock that and unpack that and figure out why it is their memory does seem to record and they retain everything they've seen. They don't find it pleasant, and medicine would like to, people in medicine want to try and understand that so they can turn it into something beneficial for people who are losing memory. From a security and intelligence standpoint, it is a really unique opportunity to begin to discover, can you administer a drug that enhances human memory for a certain number of hours? Does it have to be permanent? So rather than carrying technical toys somewhere to try and record and collect information, your brain just remembers it, which doesn't give anybody anything really to detect. So that's one potential use for it, and that is one lane of research that's going on.
Okay, so I am going to link below to this video. You should watch the entire video because this man imparts an awful lot of information that is, very important to know what they are doing, what they can do to all of us. And when you think about having control over the human race, you can literally change the thinking, the opinions, the worldview, and the behavior of a human being. You can make them violent. You can make them peaceful. You can kill them. You can do whatever it is that you want to do because you have remote control of cells that you can park anywhere in the body, and they'll sit there until they're activated. Well, that's where we're at. That is where we are at. So, CRISPR-based enhancers of DNA vaccines for COVID-19. Yes, it is true. And interesting is this. Despite the many advantages of DNA vaccine technology, the platform has yet to be harnessed for broad clinical use because so far DNA vaccines have required large doses, repeated dosing regimens, and have exhibited limited overall efficacy. Well, that sounds like our COVID-19 vaccines. mRNA technology gave us the first COVID-19 vaccines. It could also upend the drug industry. Time magazine. You know, mainstream media does report the truth on occasion. You know, bacteria developed clustered repeated sequences in DNA known as CRISPRs that can remember dangerous viruses and then deploy RNA-guided scissors to destroy them. In other words, it's an immune system that can adapt itself to fight each new wave of viruses. Just what we humans need. Now, the big MSM outfits, they may tell you a little bit of truth, but they don't really do the deep dive. Wired, could CRISPR be humanity's next virus killer? So, it has been published. It's unfortunate, you know, that so many people are refusing to take in information. And while they're so programmed, you know, to do the regurgitating of what mainstream media tells them, they stay away from the information that could lead them to more information that's, well, pertinent to their own life and their own health. But mainstream media is reporting it, some of it. New genetic method of using CRISPR to eliminate COVID-19 virus genomes in cells. Could CRISPR be the next virus killer? Yeah. Okay. The gene editing CRISPR is owned by a few multinationals. Yeah, New World takeover. And it's Dow, DuPont, chemical multinationals. Bayer. And, sorry, hold on. I apologize. I was interrupted, so I can't remember exactly where I left off. But, yes, multinationals, corporations, they own the patents to this gene editing, to the CRISPR technology. And as I have posted in another video just recently, the Supreme Court ruled that anything that is injected into you, foods that is man-made, not natural, is whoever owns the patent owns you, the food. Well, none of this is even reasonable, but this is the way the world is going. So, we have now very evil multinationals and corporations owning seeds because they have the genetic, the patents on the genetic modifications of foods. Do they own the foods that they have genetically modified? I could see the Supreme Court ruling that they do. You know, it's only been a few people who have decided to alter life itself. They didn't ask for our consent. We have not been a participant in any of these decisions. I don't like the fact that my food has been greatly altered, genetically modified, And they have used CRISPR, the technology, to alter that food. I want real food back, but I'm not going to get it back. Yeah. Why gene editing is the next food revolution. New technique. A new technique. Now, have you noticed our food looks rather like the produce that you buy? Tomatoes and apples and whatever it is. Looks very uniform, without any blemishes, or almost perfect. Well, that's the result of genetic modification. That's the result of CRISPR technology. It's already in our foods. But can CRISPR help us stop wasting so much food? Well, how can that happen? Boise, Idaho firm that introduced 2 generations of potato using RNA interference to block gene expression that induced browning recently announced that it has licensed CRISPR technology to literally extend shelf life of vegetables, of fruits, and to have no browning on potatoes. The CRISPR technology for potatoes, that might have been the first, I'm not sure, but that's been used for years and years. Using CRISPR to design superior foods, mushrooms that don't brown, apples, potatoes, non-browning mushrooms, potatoes, apples, just the beginning. Tomatoes, pineapples, wheat, pears, cucumbers, grapes, cherries, mangoes, and more. Rice, soybeans, birch trees, tomatoes, carrots, peanuts, everything. Everything is being genetically modified. And we're eating it. Genome editing in potato with CRISPR. It has been going on for a long time, and I posted on Kafka Winston World a man who, I believe, developed the CRISPR technology for potatoes, and then he came out with the truth, and he was quite remorseful about what he had done to the potato. I tried to find that information. Any information that you find, you might want to download, because very difficult to find out. You know, years come, you try to do that search again, you might not be able to find that information anymore. They are cleansing the internet of all information that could lead a human being to know the truth. Ghana scientist turns to gene editing, CRISPR, to improve the sweet potato crop. New Scientist, 2018. CRISPR gene editing is not quite as precise, as safe as thought, and as safe as thought. That's right. Alan Bradley of the Wellcome Sanger Institute in the UK. He did studies, and Bradley's team found that around 1/5 of cells, CRISPR caused deletions, rearrangements, more than 100 DNA letters long. These surprising changes are sometimes thousands of letters long. It's a cancer risk. We need to understand more before rushing into human clinical trials. Guess what we're living? That human clinical trial with the COVID-19 vaccine, COVID-19 injection. And the Atlantic, the CRISPR baby scandal. Now, this is the technology that they'll create designer babies. But I find it interesting. They came after this Chinese doctor who did this experiment with twins, with the CRISPR technology, the CRISPR-edited babies, the designer babies. And here, The Atlantic, oh boy, the hypocrisy of this mainstream media is sickening. But he didn't address an unmet medical need. Oh my God. But they're not calling out Big Pharma that's injecting billions around the world with this CRISPR technology. The actual editing wasn't executed well. It's not clear what those new mutations will do. New mutations. Well, that's what they're finding. Those who are actually doing the studies, they're reporting that it may target the gene that they want to target in your DNA, but they're not talking about the unintended consequences. And that's a big problem. Informed consent? Oh, please, Atlantic. You don't discuss informed consent at all with this COVID injection. Cloak of secrecy, that's the game. Okay, so dangers of CRISPR. Stop playing God with human genes. In the words of one of its inventors, CRISPR makes the human genome, quote, as malleable as a piece of literary prose at the mercy of an editor's red pen. That's the cell that, well, when somebody has remote control, they can edit your DNA just like an editor's red pen. So, a team of researchers at the Francis Crick Institute in London used CRISPR to edit 18 donated human embryos. The purpose, they claimed, was to study the role of a particular gene in the earliest stages of human development. Unfortunately, around half of the embryos contained major unintended edits. It could lead to birth defects or medical problems like cancer later in life. Think about all of the people who have taken that injection. As one genetics researcher put it, you're affecting so much of the DNA around the gene you're trying to edit that you could be inadvertently affecting other genes and causing problems. Fyodor Urnov, a professor of molecular and cell biology at Berkeley, said, "There's no sugarcoating this. This is a restraining order for all genome editors to stay the living daylights away from embryo editing. Well, using this CRISPR technology, stay away from it. It's not just embryo editing, but this CRISPR technology is being injected into adults all over the world. And there are many dangers, ethical and safety concerns, and enormous possibilities to further advance the human health and well-being. But it's also the case with all of this new technology. It can be used for good, or put it in the hands of evil people, it could be used or misused. And the unforeseen consequences, which, well, most don't even care about anymore. But what about the potential misuse in an evil way to control people? And here, danger posed by CRISPR. This was March 11, 2021. Just a couple of weeks ago. Gene alteration. To develop the perfect human being who has everything right going for them. You'd get a human package that is intelligent, beautiful, or handsome, physically fit and attractive, perfect to the human eye. When gene makeup is altered, then it would alter the appearance or functionality of a person as they would with the original gene. The research has been expanded to allow the treatment and elimination of genetic diseases such as Down syndrome and cystic fibrosis, but like any item made for good, this is what the warnings are. Any item made for good, there has to be someone who would want to use it for an ulterior motive. CRISPR suggests that they are developed from RNA, used as a tool to spread a deadly virus if placed in the wrong hands. Injecting a sample size of people is enough to spread it on a global scale, and that's what a lot of scientists are now saying. This injection of this technology is the tool for genocide worldwide. It alters the genetic makeup. These changes are transferred through generations. This could worry many, especially if the process did not go as planned or measurements are done wrong, causing huge unaccounted for defects in the larger public. We could start seeing medicinal resistance or mutations that would be difficult to control. So, new study points to unintended consequences. This was back in 2018. There's so many warnings out there about this technology. Research conducted in 2012, University of California, Berkeley, paved the way for using this CRISPR technology to genetically engineer animal and human DNA. Scientists soon began touting the gene editing tool as a marvel, with the potential to cure and prevent a wide variety of diseases. But researchers, yes, Bradley, again, the CRISPR to alter sections of DNA in genes by cutting at specific points and then either deleting genetic information or adding new material at those sites. The technique can have negative consequences if it accidentally modifies DNA researchers had not targeted. Frankly, I think a whole lot of the world's population has already been altered, because just because they keep coming out saying this is new technology and, oh, we're doing this experiment, we're doing this study, and no, I believe that this technology has been in the works for a long time. We have never seen behavior like we are seeing now. People who are literally just, they've lost their minds. They are acting in ways that show us they have no interest in protecting their own health, their own life. They sit back. Oh, I got posting on Facebook, I got the vaccine. They're all happy about it. You have influencers in social media posting these stupid-ass videos about how great it is to get vaccinated. And when you just try to talk to somebody who you've always understood as reasonable, they will shut you down immediately, have no interest in finding out the dangers of this new